Hi, my name is Beatrice Mihalache, and I'm an undergraduate at UCLA studying biophysics and data science, and the title of my project is Pacbio sequencing data analysis on recombinant bacteriophage libraries designed for targeting gram-negative bacteria. So for context and importance, phage-based therapies are a developing treatment for antibiotic-resistant bacterial infections. And effective treatments require specific phage-binding protein and target bacterial surface protein interactions. And one of the ways to find the phages with the specific binding capabilities is to construct large libraries of bacteriophages and then isolate the specific binding ones. So our lab did this library construction, and the way they did this was first constructing reference libraries of the G3P protein, and this protein is the specific protein that binds to the bacterial surface. So for one of the reference libraries, which consisted of 18 members, 5 oligos were used during the Gibson assembly process, whereas there were also a 43 and 200 member reference libraries constructed with 4 oligos during the Gibson assembly process. And then I was given the bacteriophage sequences derived from these reference libraries to analyze. So the project goals were to evaluate the efficiency of assembling the libraries, as well as the diversity, and create a visual representation of the sequence data. So this is my overall pipeline I created, and it resulted in a force-directed diagram that looks like this, and I'm going to break it down. So the first thing I did was isolate the G3P protein from the large bacteriophage sequence. And the way I did this was I checked for the presence of the end of the protein, and all sequences without this were discarded as they were deemed invalid G3P protein sequences. And then I continued to modify the sequences until an amino acid sequence was generated from the codon sequence. And this already gave insight into the efficiency of the library assembly, as the 18-member derived library had a clearly lower quality of G3P protein results, while the 43 and 200 member had better results, indicating the number of oligos used during the reference library creation is important. Then I've isolated the binding sequences in the libraries such that they could be highlighted in red. And so our lab, through experiments, determined which sequences bind, and then I found these specific sequences in the data set. Then I did a blast analysis by creating local databases of the reference sequences to compare the protein sequences to, and I formatted the results to be easier to analyze, as in the graph, instead of including all the relationship between the protein and all the reference sequences, I only wanted to highlight the closest 3 reference sequences in similarity. Then I checked if there were repeats created by doing a calculation called the Levenshtein distance, which checks for the number of differences in 2 sequences. And for this, I used UCLA's Hoffman 2 cluster supercomputer to do the calculations on. And then I removed repeats, and there was a varying number of repeats across the different libraries removed, also indicating certain things about the quality of the library and the thermodynamic favorability of the G3P proteins created. So now I'm going to break down the visualization. The first thing I had to do was decide on the type of graph, and I chose a force-directed graph where the nodes start off randomly placed, and then a force governs the placement of the nodes, and the force I chose was the E value. So a smaller E value from the blast results indicates a closer placement to the reference sequence. And then there were the 3 minimum E values were used for the relationship depicted. And here's the simulation again. And then the other thing I had to do next was normalize the data. So there was a large disparity of the E values. And so the first normalization I did was a logarithmic normalization. And then the goal was to map the minimum E value in the data set such that it was right on top of the reference sequence. So then the next calculation I did was the sum here. Then since I had to fit the graph on my screen, I shifted the coordinates such that the minimum X was on the left side of the screen and the maximum X was on the right, similarly for the vertical. And I solved the system of equations to do this. Then more features of the graph. So the blue nodes represent the reference sequences, while the red nodes represent the sequences experimentally found to bind well to the gram-negative bacteria, and then the orange nodes are the rest of the G3P protein sequences. Then the force governing the placement, like I said, is the E-value from the BLAST result between the reference and protein sequence. And then the darkest edge, so there's 3 edges coming from each orange G3P protein node. So the darkest edge represents the reference sequence closest in similarity, while the other 2 are the second and third. Then this is the final graph created to represent the 18-member library. So this is 5% of the data represented. And clearly you can see that certain reference sequences, like these 2 in the middle here, were used much more commonly than others, indicating something about them being thermodynamically favorable. And you can also see natural clustering arising in similarities between the G3P proteins, as well as you can also see that the binding sequences came from different reference sequences. And you see similar things for the 43-member graph, and then as well as for the 200-member library represented, you see how the binding sequences come from all over. So conclusions is that the pipeline successfully analyzed a large library of bacteriophages to determine efficiency of assembly methods. And then for this case, we were able to determine differences between the library generation. And then in the future, this pipeline can be adapted to become more generalized to other protein libraries. And then also a nicer user interface can be created. So these are some of my key references. And then I'd like to thank Stephen, my graduate student who I worked with, Shelby and Beto, and my PI, as well as everyone else in the Chen Lab. And thank you to the UCLA's Hoffman 2 shared cluster. Thank you for listening.